So welcome to today's Shore online seminar on building blocks for reproducibility. I'm Julie Goldman, Research Data Services Librarian at Countwide Library. And as a note, this session is being recorded and you will receive an email when the recording and the slides are available. First, let me start off with some services and resources that may be available to you. The Publishing and Data Services team at Countwide Library offers support for manuscript writing, copyright, research impact, data management, and bioinformatics. Additionally, the Longwood Medical Area Research Data Management Working Group has a website full of resources and information. This group includes members of the entire LMA community, and the group has created this website as a portal for learning about data management and related topics. And I suggest you bookmark this website as it has lots of information to help you manage your data, and you can find all the recordings from these webinars on this website as well. Okay, so let's review today's learning objectives. Hopefully you'll understand the important impact of creating reproducible research, establish a reproducible workflow within the context of an example, and identify services and tools available to support reproducible research. So to start, I like to define research data management, especially since managing data is an important part of research reproducibility. And research data management concerns the organization of data from its entry to the research cycle through the dissemination and archiving of valuable results. It aims to ensure reliable verification of these results and permits new and innovative research built on existing information. And why should you care about research data management? Well, there are benefits for yourself, other researchers, and science in general. You will thank yourself by documenting your systems so you don't have to rely on your memory. Data management facilitates team transitions by allowing everyone to focus on the research instead of spending time trying to decipher or find project information. Data management also enables you to conduct analyses effectively when collaborating with others. Anyone can check and verify your research results when there is well-documented reagents, protocols, data sets. And managing data helps ensure data is findable, accessible, interoperable, and reusable. And additionally, there's a movement among sponsors to require the data sets that underlie research funded by the public be made available. So we want to make sure we are being compliant with university and school policies, funding organizations that require a data management or data sharing plan, and journals that require you to submit your data along with your article. All right, so what problem are we going to try to solve in today's very brief overview of reproducibility? And the comic on the slide highlights a common issue we see in research, which is the availability and access to all the things you need to continue or reproduce someone else's work. So it's important to make the distinction between reproducing something and replicating it. And there are many ways to define reproducibility and replicability. So here I provide two basic definitions. Basically, reproducibility is where someone uses the same materials and methods of the original researcher to get the same results. And replication is where someone collects new data and new methods but arrives at the same scientific findings as another study. So today we are focusing on reproducibility. And I want you to think about why reproducibility matters to you. So the question is, have you ever had problems reproducing your own or someone else's research? And if you have never tried reproducing a research project, you can also think about trying to, say, reproduce a recipe or a craft. And I have a quick poll. So take a minute to think about this question. Great, so almost everyone has participated in our poll. And I want to share the results, so if you haven't voted, quickly do so. Great. So hopefully you can see the results from our quick poll from our group. And we have about 30 people on the call, and you can see the majority of you have said that you have issues reproducing both your own and someone else's work. And this is not surprising. So let's look at a longer sample size. So in this 2016 Nature News article, 500 scientists out their thoughts on reproducibility in research. It's overwhelmingly across people have trouble reproducing someone else's work and their own work. And when asked about the factors that can sources of issues of reproducibility include methodology, statistical and reporting practices, structural and organizational practices, and also rarely intentional scientific misconduct. So from this, we see there are many factors that are not easy to address or will take time to change, such as those organizational practices. But some of these factors related to data sharing and management can be addressed. So like we saw in the opening comment, things you need to continue or reproduce someone else's work. So we are going to focus on a few best practices that you can start code data. And in order to achieve reproducibility, as included in our computer codes to run the analyses, must be provided. So we see here that reproducibility is a spectrum from the publication only, which is not reproducible, to an article that includes links to the executable code and data for someone to reuse and reanalyze. And this will ideally be fully reproducible and what we call the gold standard. I find it too important to provide this reminder when I talk about data management and reproducibility. So there are a lot of steps and processes involved to meet this gold standard. So the takeaway is to focus on integrating 1 or 2 new practices into your workflow or research, and this will immediately make your research more reproducible. So overall, research isn't being effectively managed or made reproducible. Much of the time, the workflow and the processes aren't reproducible. The findings, such as data and code, aren't managed effectively. And as a result, we all suffer. So here, let's cover why reproducibility is important. Science is incremental, meaning it is a series of steps that builds on each other. In order to build on previous work, you have to understand that work. For sound research practices, we want to ensure what we find is correct. So reproducibility helps us avoid self-deception when you may deny the significance or importance of opposing evidence. Reproducibility also helps newcomers get started or pick up a project. And if you are new to the field, you can more easily learn the methods by reproducing someone else's work. And reproducibility increases research impact, visibility, and quality. And to that last point on research impact and visibility, you can think selfishly about reproducibility. By sharing all parts of your research, others can reuse and extend your work more easily. So you may even find interesting collaborations and future research projects out of this. And of course, remember, future you is your work. So as shown on the slide, in Publisher, you receive a citation for your data, and that data set can be cited in other places, in other journal articles. And therefore, your visibility and impact is increased when other researchers view or reuse that data set. But also remember that you or someone else cannot simply reuse a data set. You need all the appropriate metadata and documentation about that collected data in order for it to be reused. Perhaps in order to integrate it into another data set or compare it to other data. So what do we need for reproducibility? And hopefully you will see some past webinar topics in these key practices for reproducibility. So first, be organized. Organize your projects so that you don't have a single folder with hundreds of files, and use tools to help you stay organized. Keep documentation through the entire project. Clearly separate, label, and document all data, files, and keep track of operations that occur on data and files using version control. Use automation to your advantage to reduce human error. So design a workflow as a sequence of small steps that are glued together with intermediate outputs from one step feeding into the next step as inputs, and use scripting to create automated data analyses. And finally, disseminate all parts of your work. This includes data, code, and materials. Additionally, publishing is not the end of your analysis. Rather, it is a way towards your future research and the future research of others. So I'm going to introduce the concept of the recipe for reproducibility. And who has ever followed, say, a baking recipe? Isn't it much like following a scientific protocol? And this recipe includes data and materials that can be formed into information, which can be presented in many different ways, and then it's consumed as knowledge by others. So this is our recipe. And what should be in this reproducible project recipe? Your recipe should include those key practices and all the details needed to go from the data to information every time. So let's match those key practices to 4 steps in our recipe. So 1, providing data quality assurance through documenting how data was collected, validated, and organized. 2nd, including all contextual information needed for discovery, access, use, and reuse. This includes describing data and providing all relevant documentation. 3rd, include code and scripts associated with analyses and results. And 4th, submission of data and all related research products, such as protocols, materials, and code, to relevant open repositories. And how should this information be packaged? You could package all this information into a README text file. So a README file contains all the metadata or information about the data that you need to remember what your data is, and others need to understand what your data means. So create a plain text file for each distinct data set. And for each of these files, you should include some basic project information, title, contributions, grant information, contact information, all locations of where data live, including any backups, and published data in repositories, useful information about the files and how they are organized, especially explaining file naming conventions and abbreviations. And for these READMEs, ask yourself a few questions. So what experimental and analysis-related information is important for you to keep track of? Would a new project member be able to step in and know how the data was created? Would they be able to reproduce the analysis? And would they be able to follow what you've documented? So on the slide is a very nice example of a README template from Cornell. And you can find this and other README guidance on our website. So documenting your metadata is key to reproducible science. Ideally, anyone who has your data and the documentation can recreate or reproduce the information you originally created. So much like having all the information in a recipe to bake the same cake every time, the same details should be included in your README files and or your protocols for others to reproduce your work. Okay, so focusing on these 2 areas, reproducibility and data management. You can't have reproducibility without proper project and data management. So with this overview, I hope you can see how intertwined all these concepts, data management and reproducibility are. Data management and its related activities are fundamental for supporting findable, accessible, and reusable research. And data management principles ensure that gold standard for reproducibility. Okay, so for the last part of today's seminar, I'm going to walk through an example of a situation where all the pieces of a research project were adequately documented and shared. So this example is for a single-cell RNA sequencing study, but I won't focus on the study area. This is really about seeing if we can find all the parts of a project in order for someone to get the full picture of the work and potentially reproduce a study. So our starting point is a preprint, and I want to see if you can locate all the supporting documents you need to reproduce this study. So I would like you to take a minute or two to see if you can locate the published peer-reviewed manuscript, the code, the data, the methods protocol, a README file to the data files, And maybe there are some other things you can locate related to this study. So I've put a short link that will lead you to the preprint and see if you can find the things that I've listed on the slide. And as you find them, you can indicate on our next poll if you have been able to locate. So I'll give you a few minutes to work through this example, and then we'll go through it together. Okay, great. We'll come back together and keep working through this example if you are. Looks like most of you have successfully found the elements, so that's great. So let's walk through this example together. Oops. Okay, so we start at the preprint of this study, which is published in bioRxiv. And this preprint has a DOI and can be shared and cited in itself. And we see once this article went through peer review, it was published in Nature Communications. The DOI is right at the top, and we can follow the linked DOI to this published article. So here we are at the published article in Nature Communications. This is an open access article that was published in 2018. So we want to find all the supporting materials for this article. And so we can scroll to the end of the methods section just before the references start to find both a code availability statement and a data availability statement. And these statements include information and links to where one can find the supporting materials for this article. So we can find and explore the code on GitHub. Whoops. And we find the data set in GEO, the Gene Expression Omnibus. So GEO is an NIH and CBI public repository that archives and freely distributes microarray, next generation sequencing, and other forms of high throughput functional genomic data sets. And a series record defines a group of related samples and provides a description of the whole study. So this series record may also contain tables describing extracted data, summary, conclusions, or analyses. And each series record is assigned a unique geo-accession number. And so this geo-series record contains experimental data from our single-cell RNA sequencing using a new protocol for something they're calling MCSC-RB-seq. And from this page, we can find descriptive information for this series record. So this might have been a little harder to find, although some of you said you found some documentation. But from that geo record, we can also get a text file that contains all the data and the metadata for the genome samples and series included in the data set. And this was found under the download family section on the previous geo-accession record. And so there are a few bonus places we can find related to this project, linking from the geo record. I'm just going to point them out, but these are some NCBI bioinformatics databases you may be familiar with or already use. So from that geo record, we can also explore the project DNA sequencing data and even more data through the SRA run selector. And this is on the sequence read archive. And also find the bioproject entry to see all the research products that have resulted from this project. So from here, we see that there have been a number of SRA experiments, a number of biosamples or source materials that were used, our one geo data set, and the one publication linked in PubMed. Okay, so if we go back to our article and back to the data availability statement, we see there is one more reference we can follow, and that is to the protocol that is shared on protocols.io. And while the statement itself only has the title, which we could find by searching protocols.io, there is also a nice citation to it in the references, number 29. So we can use this DOI to find the protocol. And this is a very nice example of a protocol with great documentation and links to our research article. So it looks like people were able to navigate through this example to find all the related research pieces. This is a good exercise to work through on any article or data set you come across. Try to follow the links and connect the dots between articles, data, and other supporting materials. As I mentioned at the start of this webinar, sharing and publishing data sets, code, and documentation is now generally required by funders and journals. But through this example, you can see how making all of these elements available, one would ideally be more successful in following or reproducing this study. But I just want to make it clear that just because all these elements were openly shared, it still does not necessarily mean the study is reproducible. There are many additional factors that need to be considered. So you must ensure that the underlying raw data are made readily available. All software and tools must detail the appropriate version. Underlying analysis tools are made readily available. The code, data, and materials are transparent and adequately reported. And this last point highlights the lesson from our recent protocols.io online workshop, that writing informative methods is more important than the platform itself. But the overall goal is to encourage the community to consciously choose open tools to increase interoperability and sustainability of your research. So in our example, you can see how using GitHub, Geo, and protocols.io to share pieces of this research study illustrates this. So to close, here are the building blocks for reproducible research. Plan for reproducibility before you start. You should create a data management plan at the start of any project. Organize productively and keep track of things. Track changes using version control and document everything in README files. Incorporate automation if possible to enhance your workflows and reduce human error. And report your research transparently. Share your results, methods, materials, documentation, data, and code. Any research products you use or reproduce or produce in addition to any article. And all of these activities together will help you make your work more reproducible. Great, so I hope you have identified some steps you can take to increase the reproducibility of your work. I'll just go through these takeaways from today's session. So reproducibility is when materials and methods are used to get the same results as a previous research study. You should plan for reproducibility before you start your research project by creating a data management plan or writing a study plan. Organize your projects, track changes using version control, and document everything in README files. Find ways to reduce human error through automation of scripts and document all software versions and code used. And share and license your research products, including data, protocols, materials, and code through supported repositories to enable discovery, access, and reuse. So please see this website for more on all of the topics discussed in today's webinar, and feel free to submit any questions and feedback using the online form. We have 2 more June seminars that highlight 2 tools for data and project management and organization. So sign up at the link on the slide. So thank you for joining the session today. The recording and the slides will be posted on our website. I would appreciate your feedback on today's session. So please let me know if there are any specific topics you would like to see covered in future sessions. So please fill out the survey at the link provided on the slide.